So my name's Sawyer. I'm from the University of Queensland, and today I'm going to share with you some of the results from a small field trial that we conducted last year along with Myrtle, and we were testing our double-strand RNA. So before I start, I'd just like to acknowledge the traditional owners of the land we're coming today, but also of the land on which this research was conducted, and I pay my respect to their ancestors and descendants who continue cultural and spiritual connections to country, and I recognise their valuable contributions to society. So some of you might have already heard about double-strand RNA-based biopesticides, but I'll give another introduction in case some of you haven't. So basically, it's an environmentally friendly approach with an alternative chemical pesticide. It's quite a new technology. So it involves spraying double-strand RNA that's specific to in this case, Myrtle onto plants. And this is highly specific. We can use bioinformatics to make sure that the RNA is only targeting myrtle rust and no other beneficial organisms in the environment. And I have a slide coming up that sort of shows how we can do that. So it doesn't leave any chemical residues on the plant, or it doesn't contribute to any chemicals running off into the environment. We don't need to modify the plants. There's no genetic modification required. It's a transient approach. And we can load the RNA onto carriers to protect it from degradation. But I will show that the double-stranded RNA is actually quite stable on metastases, interestingly. So if you look at, I'm not sure how to use this one. If you look at that schematic, that's sort of showing what the approach involves. So we spray the double-strand RNA onto the plant. In the case of myrtle rust, we found that the plants can be healthy or infected. So the RNA can have both a preventative and a curative effect. The double-stranded RNA is actually taken up by the rust fungus itself. And inside the fungus, it triggers this natural defense mechanism. It's a highly conserved mechanism known as RNA interference. And this involves the organism or the cell recognizing that double-stranded RNA as being something foreign. And so there are enzymes that actually target it for degradation. So the RNA is actually chopped up into small interfering RNAs, and they are then used as templates to find any homologous RNA. In this case, it's the messenger RNA. And so this pathway actually stops essential proteins from being produced in the pathogen, and so it inactivates the pathogen and prevents it from infecting the plant and causing disease. So this is what I was talking about. This is actually a recent paper published. This is led by Steve Fletcher, who's a bioinformatician who's been working with us, and he can actually design our double-stranded RNA so that they're highly specific to one species. This is actually a nematode example. We've also been looking at root knot nematodes. So in this case, we wanted to target 4 different species of Myeloidogyne, which are the root knot nematodes. And so he actually made these sequences using a bioinformatics pipeline to make sure that they would only target these 4 species and not target C. elegans, which we used as a sort of off-target in this case. And it worked really well. Those 2 graphs, he had 2 different double-strand RNAs of different stringencies, so we can change that if required. And if you look at the bottom sets of graphs, that's showing the efficacy. So we actually needed very small amounts of RNA. It was very effective against these 3 species that we tested. And then the graph on the right, we also looked at C. elegans. And importantly, the RNA targeting the root knot nematodes didn't affect C. elegans, which is great. So if you want to look into this, you can have a look at the paper, but I just wanted to show you that that information is now available. And also, there's now a product on the market, a double-stranded RNA-based biopesticide. This is available in the US. It's called Calantha, and it targets Colorado potato beetle. It's been released by Greenlight, and they have a lot more products under development at the moment. So it is actually now becoming a reality, which is quite exciting for us. But back to myrtle rust. So you've probably heard about some of our research. These are two papers that we've published. We've found that we can target very essential fungal genes. So we've been synthesizing RNA targeting beta tubulin and also the translation elongation factor and CDI. And we've shown that when we apply the double-strand RNA, it can both prevent and cure myrtle. So the paper on the right was our liquid paper showing when we applied double-strand RNA to plants at various stages of infection, it actually helped improve their recovery from disease. They still did get infected, but you can see the image on the right is the plants that were treated with the double-stranded RNA. These photos were taken 6 weeks after inoculation with the pathogen, compared to the images on the left, which were the control plants that weren't treated with the RNA. So you can see it really did help the plants recover from disease. So this was all done in the glass house, and this was actually all on Syzygium jambos, which is not a native plant in Australia. But it's very susceptible to myrtle rust, which is why we're using it. But we wanted to take this into the field and test it on a native plant. So that's kind of what we did last year. We did a couple of very small field trials at Australian Native Products, which is a lemon myrtle producer. They have 2 sites, but we went to the Channon in New South Wales. This is a site that's very conducive to myrtle rust. It's very close to Lismore. If you know Lismore, it rains a lot, and so there's a lot of disease around. And I'd just like to thank Kirsty Langdon from AMP, who helped us conduct the trial, and also Louise, Alistair, and Sebastian, who were with me in the field doing all the work. So thank you very much, you guys. So we did 2 trials. The first one was sort of a pilot. We didn't really know how we were going to do it. We were very ambitious at the start. We had 8 different treatment groups, which we realised was actually a bit too much. But anyway, we had a go at doing this. So we had a non-spray control, water control. We had a non-specific RNA control, which was just green fluorescent protein or GFP. And then we had our 2 myrtle rust specific targets, so TEP and tubulin. And then because we're in the field, we thought, oh, maybe we do need to protect the RNA from degradation. So we wanted to test bioclay as well. So that's where we load the RNA onto a clay particle. And it's been shown in previous studies that this can actually improve the stability of the RNA on the plant. So we had a whole another set of treatments with the RNA loaded onto the clay. We used a moderately susceptible variety. That's because this was the variety we were using in our glass house, so we had done a little bit of work on it. But the only problem was that this sort of restricted us in the number of trees we could use. This was actually an old DPI site, so it had a lot of different clones planted on it. So all those squares are different genotypes, different clones, sorry. And so the blue ones were this one moderately susceptible clone that we were testing. And unfortunately, all the trees were right next to each other as well. So you can see in that photo the different coloured tape. So unfortunately, our treatment groups were all next to each other. And we did use a barrier when we were spraying, and we were using just hand sprayers. So we weren't too worried at the beginning, but then we realised there probably was a bit of spray drift. But anyway, it was all a learning process. And so we actually tagged individual branches. So we tagged four healthy branches because we wanted to do a preventative and a curative experiment. And then we tagged 4 infected branches on each tree for each treatment group. And we applied about 45 milligrams of RNA per tree. And so I'm still trying to figure out the best way to present this data, but I just thought I'd sort of show an overview. We assessed disease on different aged leaves. So we had like older leaves, which are the blue fully emerged at the bottom of these graphs, newly emerged, which were the next set, and then the youngest leaves at the top. And we also noted if there were stem infections, so that's captured on that graph as well. Even though this site has a lot of myrtle rust, at this time, we actually didn't get a lot of infection, which was a bit of a problem. Our controls didn't get super infected. So it's quite hard to see the effects of the RNA. But at the 4-week time point in the curative experiments, the TEF and the tubulin in both the naked and the bioclay treated plants, the leaves, the youngest leaves were still healthy, which was quite promising. But then by 9 weeks, the leaves were more infected. But anyway, it was still an interesting trend. This is the 9-week assessment, and this is just showing the disease coverage on the youngest leaves for both the preventative and the curative experiments. And so in the preventative experiment, the youngest leaves of the tubulin spray plant still did have a significantly lower disease, amount of disease than the control, which is the GFP. But like I said, the water control didn't really get very infected, so that kind of affected the results a little bit. And in the curative experiment, we did see a trend with the TEF, but once again, we did have some issues with the control, and the beta tubulin didn't look as promising. Interestingly, the bioclay didn't really look so great. For the curative, we weren't really expecting it to have much of an effect because we wanted the RNA to get in straight away anyway, but in the preventative, it might have helped to have the RNA sort of gradually released onto the leaves while they weren't infected. But it's possible there was some latent infection because most of the trees were infected, so possibly what we labelled as preventative had already been infected anyway, so that might have been why we saw those results. And so this is just another way of me trying to show the data. This is just the different symptoms. Sorry, I don't really have photos for each type of symptom. I'm assuming most of you have seen a lot of myrtle rust, so you probably know what it looks like. But we kind of sort of scored based on whether the leaves were healthy, if there was flecking, pustules, lesions, or necrosis. And we gave a kind of a percent, like 50% of the leaves were covered in pustules, for example. So that's kind of how we did the scoring. But there was quite a good trend. So if you look at the beta tubulin and the TEF, this is all just the naked RNA results. There were more healthy individuals for those treatments and less necrosis, fewer lesions, that sort of thing. But anyway, we did want to repeat it. But another interesting observation, we checked for the RNA on the leaves, and we found we could actually detect intact RNA 9 weeks after spraying for both the naked RNA and the RNA bound to the clay. So this is really interesting. We didn't expect double-strand RNA to last so long. This is not quantitative results. We still don't know how much was on there, but it's showing there is still something intact, either on or in the leaves. But that was kind of an interesting observation. Anyway, we went on to do another trial to try and make up for some of our mistakes in the first trial. So this time we only did 4 treatments, but we had a lot more replicates. So we just focused on the double-strand RNA, because that seems to be more effective. So we had 4 trees this time per treatment, and this time we tagged 20 branches per tree. And instead of trying to differentiate between healthy or infected, we just scored all of those branches at time 0 before we sprayed. So we had like a baseline. And so then we could compare afterwards to the initial kind of disease load. And we sprayed 180 milligrams of RNA per tree, so a bit more this time. And we also used a more susceptible commercial variety. So there were actually more trees available to work with. So that's kind of why we did that. And this is just sort of showing the block design that we used. So we had buffer trees this time, so we weren't as worried about the spray drift. And so this is sort of, again, we had a lot of data, and it's kind of hard to distill it all, but this is sort of just giving you the overview that there was significantly less disease on the TEF spray tree, which was really exciting. The beta tubulin was also lower, had a lower amount of disease compared to the GFP control. But once again, the water control had a bit less disease than the GFP control, so there was no significance between beta tuber and water. But it's still quite promising, and we will hopefully, if I get some more funding, be able to repeat this field trial with some more replicates at a different time of the year maybe, and see whether this sort of trend holds up. So yeah, we still have a lot of questions remaining. And also I should have said in that last trial, we only did the disease assessment at 12 weeks. Based on there was a lot of rain, which prevented us getting out to the site. So if we do it again, it'd probably be good to do an earlier time point to check the symptoms. And we might also want to do another dose, like another spray, like a month after the initial spray, for example. So we want to check that sort of thing, like the dose, whether we need to increase the frequency of the spray. We want to see if there's systemic protection. So if we spray some of the older leaves, are the newer leaves also protected, or do we actually need to spray the leaves that are going to get the rust spores on them? And another thing we're interested in the moment is, can RNA be used in combination with other controls? We don't see it being used on its own solely. We see it being used as part of an integrated disease management plan. So we would like to test some other things, including fungicides, but other, maybe even biocontrols, who knows. And one of the remaining questions we have is, why is the RNA so effective against Australasian TDI? We're working on some other pathogens where the RNA isn't as effective. So this is kind of an interesting question we're looking at the moment. And in terms of myrtle rust, conservation management, we see that RNA sprays could have the potential to protect native plants in nursery environments, maybe also in ex situ conservation. It could be used to protect plants coming out of tissue culture, out of the seed banks, plants being used in revegetation programs. That's kind of where we see RNA potentially having a role in the management of myrtle rust. So that, I'd like to thank everyone who's involved, all of my industry partners for their support, and thanks for listening.